Great to have you all as the episode 1 of the EduSearch Series, an exciting new initiative by RSG-India in partnership with EMBL-EBI. I am Jatin Joshi, the RSG-India president, and I am thrilled to kick things with you today. Most of us have used the protein data bank, whether to visualize protein structure, explore binding sites, or fetch data for modeling. But have you ever wondered how all that rich structural information comes together, who curates it, and how it's made accessible? That's exactly what today's session is about. So we are exploring PDBe-KB, the protein data bank in Europe knowledge base, and hearing directly from the people behind it. Expect insights into this data pipeline, how to navigate the resources efficiently, and tips to get more of your structural biological work. So before we jump in, let's introduce our organization, RSG India. So as you guys know that RSG India is a part of the Student Council of ISCB, which the motto is your local window to the global opportunities. So here, as you see that RSC India is the student council of ISCB, and this is the triangle of formation. And as we go, that ISCB is the full form of the ISCB, International Society for Computation Biology, a recognized global organization based in US, maximizing the global impact of computational biology on science and society through scholarship, research, training, outreach, and inclusive community building. And under ISCB, there is a student council named RSG India. And then RSG India has three nodal nodes in India. And first is node in Hyderabad, second is in Kerala, and third is Karnataka. And these are the Mahindra University, NIT Calicut, and Manipal University are the nodal colleges of these areas. And here is the team members of the RSG India. And next, these are the previously conducted events in these past 10 to 12 years, which are in by RSG India. And these are some of the photos. And then these are some of the events that we conducted. So PDBe-KB. This is an event about the PDBe-KB, and today we will learn about it. And then as we go further. Welcome to the EduSearch Series, an initiative aimed at connecting the Indian bioinformatics community, some of the world's databases and resources. Through expert-led masterclass and demonstration, this series seeks to provide clear and practical insights into essential bioinformatics tools. So we are pleased to begin this series with PDBe-KB, the protein data bank in Europe knowledge base, developed by EMBL-EBI, a global leader in biological data. So let's start with the introduction of our speaker, Mr. Joseph Ellaway, and his intro is given by Akriti Jain. Akriti.
Yeah, thank you, Jatin, and thank you, Joseph, for joining us today. It is my absolute pleasure to introduce our esteemed speaker of the day, Mr. Joseph Ellaway, and he is a bioinformatician at EMBL-EBI, and he works in the team of Protein Data Bank Europe (PDBe), and his research focuses on systemic classification of protein conformational states utilizing structural bioinformatics algorithms to better understand the dynamic nature of protein structures. He has contributed to data pipelines for identifying conformational variability at the PDBe and is involved in modernized web services for macromolecular assembly, which is one of the web server we use PISA for protein interfaces and macromolecules. So Joseph holds an MSc in bioinformatics and theoretical systems biology and a BSc in biochemistry from Imperial College London. And he studied during his master's and bachelor's projects in cryo-electron microscopy and X-ray crystallography model refinement. So he's done all these high-tech works in his graduation, and in PDBe he's doing great job in combining those macromolecular structures. So his projects included X-ray crystallography modeling refinement as well as molecular biology research on neuropathic pain at the University College London. He's also one of the developers of the open source tool Ramachandran Plotter and is active in community training and outreach. And today's masterclass with Joseph will walk us through the PDBe-KB's interface, features, tools, and how this resource can be utilized for structural bioinformatics research. So I request all the participants not please don't raise hands in the middle or please type your questions in the chat box. We will have a question and answer round at the end of Joseph's session. Before we dive into our today's session, I request our faculty advisor, Professor Manish Kumar, to please open the session with his insights. Thank you, Joseph, and over to Professor Manish Kumar.
Okay, so good afternoon, everyone, and welcome, Joseph, to be part of our initiative about spreading information about the PDBe-KE. All of us who are working whether on protein structures or any type of enzyme function prediction or annotation, we always use PDB as a gold standard. But generally, we don't bother about how the data is collected, how painstaking it is, and also most of the time we are so focused in our objective that we forget to explore what other things we can do. So I hope this Edu series will help the students in getting acquainted about the different resources that is there on PDB, and they will get benefited. So thank you to the organizers for taking pain to organize such a informative sessions, and I hope all the participants will uh, get benefited from this initiative. And if you have any question, please write to the chat box. We will try to answer all those questions. Okay, so welcome to this new Edu series. Thank you.
Thank you, sir. Over to you, Joseph, and thank you for taking this session with us.
Excellent. Well, thank you so much for having me. It's a real pleasure to be here. Let me go and start my screen share. You let me know. Can you see that okay? If I make that
yes.
larger. Is that visible to everybody? Excellent. Let me see if I can minimize that a little bit. Make that a bit smaller. Okay. Well, good afternoon, everybody. Thank you so much for the introduction. My name is Joseph Ellaway. I'm a bioinformatician at the Protein Data Bank in Europe. I think I've got about an hour and a half with you guys today. So I'm going to be discussing a bit about what EMBL-EBI is more broadly, what the PDBe is, and also some of the benefits of using the Protein Data Bank in Europe's knowledge base as well. So the real aim with the Protein Data Bank in Europe knowledge base, I'll refer to it as PDBe-KB for short, is to try to create knowledge from the macromolecular structures that we have in the PDB archive, as well as the functional annotations, which we can collect, curate, and enrich. And display that to you guys on our web pages, as well as give you programmatic access via APIs and other open source databases as well. So let's get into it. There we go. Okay, that's working. Excellent. So I'll just talk about what EMBL-EBI is more broadly. So EBI is part of an organization called the European Molecular Biology Laboratory, and we're comprised of the number there says 1,800, but really we're about 2,000 people now across 8 different nationalities and 6 different sites across Europe. So there's EMBL-EBI, EBI standing for the European Bioinformatics Institute, based in Hinxton, which is just south of Cambridge on a nice leafy campus. I say nice, depends when it's raining or not. And we host a lot of the bioinformatics services, which I report for lots of different things. I'll talk about that in a bit. But the other sites we have across Europe include Grenoble, which have a synchrotron for structural biology research, and macromolecular structure determination as well. We have a site in Barcelona too, who specialize in a lot of tissue biology and disease modeling research, primarily from the wet lab side of things. But we've also got Hamburg and Heidelberg as well. Heidelberg being the headquarters for EMBL more broadly, and Hamburg doing a lot of structural biology research, and Rome as well, based in Italy. They do a lot of epigenetics and neurobiology research, again from all the wet lab side of things. So really today, I'm going to be talking about the Protein Data Bank in Europe, who are based over in the EMBL-EBI site. So the mission of EMBL-EBI, or EMBL more broadly, as I mentioned, we're made up of 2,000 people across 29 member states. So although we have 6 different sites, which are providing the research and facilities, we've got 29 different member states which are collaborating with us. In 2023, the figures were 700 publications. And in terms of the users for the experimental resources that we manage, we had almost 4,000 in that year. In terms of EMBL-EBI's research, and well, I say the research, but also the web services and data collation and distribution, per day, the number's gone up a bit since 2023, but we have about 107 million daily requests, which is a pretty mind-boggling number at this point. But the research and the data services that we provide is used pretty much everywhere across the world at this point. And we try to make as much of this work as available and open access, openly available to as many people as possible. So in 2023, we had 8,600 different course participants and conference participants from the outreach events we were doing, such as this one here today. So in terms of how the data resources work at EMBL-EBI, this is not just for the PDBe, but more generally. Scientists will begin by generating data from their research and wet lab investigations. This is where their discoveries are made and the publications are sent out for peer review and publication. They then go about depositing it with us, depending on the nature of the data, whether it be more nucleotide-based data, structured data. It will go to different resources, and they often have to do that on publication or in conjunction with their publication. And then it's our goal to try and archive and share that data with all of the other researchers as well. So we'll try and enrich it as much as possible, as well as combining, classifying, curating some of this data as well, but also making sure the raw data files are accessible to any researcher wherever you are in the world. I think someone's microphone is on at the moment. Okay. I don't know. We can do questions at the end, maybe, if we collect them all. Yeah. Okay. Yeah. So in terms of the 5th goal here on the slide, anyway, one of the goals is to try to distribute and data, so that include data enrichment and curation, as I was saying earlier. And there we go. And hopefully the goal of that is to make sure that scientists can use the data from others to try and enrich their own research, whether that be from the preliminary investigation side or meta-analyses and other big data investigations as well. So this is just a bit of an animation over here to show how the resources at EMBL-EBI have grown over the past few years. As I said, we get about 107 million requests per day now, so it's a pretty enormous number. This is across all of our sites, PDBe included. But in terms of the specific types of data that we have at EMBL-EBI, it goes all the way from the very smallest of scales. So we're talking here about the small molecules, ligands, chemicals, particularly important for drug discovery. The resources we have include things like PDBe, Chembl, metabolites, open targets. At the PDBe, we try and connect with as many of these as possible because often structures are solved with specific small molecules bound to them. So it's going to be really important and really useful to connect to those databases. In terms of genomics and other nucleotides more broadly, we have databases like Ensemble, the European Nucleotide Archive, Expression Atlas, Magnify, RFAM, Wormbase. So these will go from genome fragments to as you say, the biofunctional annotations and trying to annotate the specific genes from whole genome assemblies to more specific ones like worm base as well. Now, on the protein side, this is where I'm more involved. Resources include things like the AlphaFold database, which I'm sure many people have heard about at this point. But we've also got Enzyme Portal, Interpro, the Protein Data Bank in Europe, the PDBe Knowledge Base, PFAM, Pride, and UniProt. And these go all the way from annotating and curating protein sequence data, all the way to protein structures as well. So again, at PDBe, we try and connect to as much of these as possible where relevant. So you as the researchers can, find and answer the research questions that you need. On a larger scale, we've also got several resources for bioimaging as well. So this will include, image data of cells. So we've got things like the Bioimage Archive, which will include light microscopy and confocal microscopy data, but also the EMDB or electron microscopy data bank, and Empire as well. So the electron microscopy public image archive too. So these guys are providing data from electron microscopy and other microscopy experiments. The data files here can be pretty enormous because you get everything from subparticle analysis, so that's where you might be trying to solve a structure using electron microscopy, all the way to getting whole cell volumes as well. So the data can be pretty enormous in scale, but their visualizations are pretty awesome. And also on the genetic variation side as well. So we've got things like the COVID-19 data platform, Decipher, European Genome Phenome Archive. So this is really where we're going from looking at the genomic variation all the way up to how that has an impact on the phenotype as well. So super important and useful platforms there. And then finally on the literature and knowledge side as well, we've got things like biomodels, biosamples, Europe PMC, which is a search engine for life sciences publications, and Reactome as well. So the talk today is all about the PDBe-KB, but I'm just going to give a brief overview as to what the Protein Data Bank is, if anyone's not familiar with it. So the Protein Data Bank was an initiative started in 1971, so very much ahead of its time in terms of trying to make data openly available and aggregate it all in one place. So the Protein Data Bank in today's form is made up of 3 different sites across the world, where we've got RCSB PDB based in the US, the PDBe, so the Protein Data Bank in Europe, based in Kingston in the UK, and also PDBe-J in Japan as well. And the idea here is that we try to distribute the workload of data ingest or deposition across these 3 sites. So if you're a researcher based in North America, South America, or Australia, and you go and solve a structure, you'll be depositing that with the Protein Data Bank in the US, so RCSB. If you're a researcher and you solve a structure in Africa or Europe, you're going to be depositing with us in the UK. And then finally, if you're a researcher solving a structure in the Middle East or Asia, then you're going to be depositing with PDBe-J. That said, we sync the data across all 3 sites, so it really doesn't matter which one you're going to. You're going to get the same coordinate files, regardless of whether you're at the RCSB website, PDBe, or PDBe-J. In terms of data distribution, the coordinate files will be the same, but we try to enrich that data, display it, make it as useful as possible to the scientific community. But in terms of the core coordinate files, you'll find them all on each site. Now, collectively, we form the worldwide PDB, or the Worldwide Protein Data Bank. And this is a collection of all of the sites to agree on data standards, as well as the deposition systems as well. So, for example, the much newer the much newer MMSIF file format is standardized using the WWPDB, and it's since sort of overtaken the PDB file formats since then. And we agree on making sure that the deposition process is functionally the same across all 3 sites. So you've got the same experience regardless of who you're depositing with, so it doesn't really matter where world you're solving your structure. So specifically the protein data bank in Europe, from most people's experience, this will be a website. You can find it at pdbe.org. You'll get a redirect to a website over here, and on our landing page, we'll try and present some information on things like featured articles every month and some statistics on how many structures have been solved in a given week, and also link you to some of the specific tools that we have available. I'm going to be talking about the KB specifically, but yeah, if we had an entire day or entire week, we could probably go through every single one of them. But you can also make searches as well on the PDB archive as well. So PDB archive is the entire database of all solved macromolecular structures that we have. And you'll see a view like this on the right. So if you want to, you can follow along, or you can go and check out this website. There's a QR code in the top right-hand corner. By no means obliged to, but I thought I'd just link it there to have a look. Now, specifically on the PDBe-KB, so the PDBe knowledge base side of things, the PDBe-KB is intended to be an open collaborative consortium of structural biology and bioinformatics data providers. So the PE and the PDB archive more generally is a fantastic resource because there are often lots of proteins which have been solved multiple times, either with different ligands associated with them, under different conditions, maybe they have different oligomeric states. And searching on the PDB archive alone, well, you might be able to find certain entries that certain researchers have solved. But the goal with the KB is to try to aggregate those together so you can get a more of a collective appreciation about what are the differences, what are the similarities between protein structures given the same protein sequence. And the whole idea is to try and enrich that data in the PDB archive with additional functional annotations. There's a highly collaborative goal here. So as I said, yeah, the goal of the PDBe-KB, and I'd say PDBe more broadly, is to establish this high quality, up-to-date integrated infrastructure using hopefully what we would think is some of these innovative data analysis tools for 3D macromolecular structures to support research, both the fundamental lab research as well as the more applied research as well, and any education as well, which needs to go on too. And the general pipeline would look something like this, where we take in macromolecular structure data, whether that be the experimentally derived structures. I don't know if anyone can see my cursor when I'm moving around at all, but on the left-hand side here, I think it is visible. There we go. Yeah. So we generally take in experimentally derived structures, but these days as well, we can also use predictor models, often from the AlphaFold database, which is a really pretty incredible research tool at this point, and try to add additional information on top of those as well. So with protein structures, you get the 3D coordinates, the 3D atomic coordinates, but once you start to add on things like ligand binding sites and annotations involving those ligands, are there any variants that are within the PDB archive? So mutations there, do they have any effect on the structure of the protein or the active site? Do we find that there are different oligomeric states? So in this case, this would be a homotrimer, but does it also exist in a monomeric form or a dimeric form or a larger complex at some point? Once you start to aggregate that information, hopefully you'll be able to answer questions like the one on the right-hand side here. So maybe given some sort of mutation, will that have an effect on the protein? If this is an enzyme, will it have an effect on its function? And does this go all the way to the phenotype? Are there any disease implications associated with the underlying molecular change which is going on? Now, the PDBe-KB consortium members, there are 34 of them now, across 13 different countries. So it's a really international collaboration between partners. And they're providing us with structural bioinformatics information. And with the PDBe-KB, our goal is to collect that information and present it on our website, as well as via APIs for programmatic access, to deliver those annotations to the scientific community. Specifically, what are they providing? Well, everything from things like biophysical annotations, any small molecule binding site predictions, protein binding site predictions, domain annotations as well, evolutionary conservations, mutation effect as well. So lots and lots of diverse, valuable annotations. Depending on your particular research question, you might find several of these useful, maybe only a subset. And often they'll cover a pretty large fraction of the entire PDBe archive. And there are a few which are more specific in scope. So for example, we've got things like metal PDB and KindCore as well. Now, not every protein will be binding to metal ions, but where they are, having those additional annotations can be really, really useful when you're trying to interrogate your structure and learn about its function. So in addition to that, we also manage something called the 3D Beacons Network, which is led by one of the Bioinformatician, also one of the data leads of the PDBe, Srinath Nair. And 3D Beacons is an open collaboration again, this time though between macromolecular structure providers. And the goal here is to provide a unified programmatic access to both experimentally determined structures, so what is in the PDBe archive, but also predicted models as well. So the PDBe archive does not include every single protein structure within the protein universe, which is why predicted models can be so useful. Some of those specific data providers these days, of course, the AlphaFold database, the PDBe, that's where we're providing the experimentally determined structures. We've also got Swiss Model, Model Archive, SASDB, PED, AlphaFill, and several more as well. And all of these structures are available via API endpoints. So if you've got a particular protein in mind and you want to search for it, well, you can go via the UniProt accession. And UniProt are sort of the guys who will manage and curate the protein sequence universe. But we also support a sequence-based search as well. So if you've just got a protein sequence that you want to query, we have an API endpoint for that as well. Now, in terms of the specifics of what the PDBe-KB provide, we want to align ourselves with this idea of sort of FAIR principles. And I'm sure some of you have heard of this before, but FAIR stands for findable, accessible, interoperable, and reusable. So the findable aspect is making sure that you can go and search and locate the data that you need for your work. Accessible is ensuring that you can download, read from a website that data. Interoperable is really important. So this is ensuring that file standards are consistent, making sure that data and file formats are usable across different software suites. And this also goes for some of the applications and software packages that we will design ourselves as well. And then reusable is making sure that the data can be stored and distributed continuously. Of course, as I've already mentioned, the core goal is to enrich this data so that we can provide biological context through the development of new tools. And usually most of this will be accessible via our aggregated user proteins page. And I'll be talking about that more in a bit. Specifically, we've got pages on things like the molecular entities. So these will be the large macromolecules with proteins, DNA, RNA, as well. The specific ligands they're binding, as well as any domains they have and complexes they form. 3D Beacons Network, well, I've mentioned that already. The data as well here specifically is available via our graph database and graph APIs. I'll talk about that more in a second. That's a really, really, really useful tool for potentially designing your own queries and getting a look at data from a non-relational point of view. Pretty much all of our software components are available as open access software tools. We put as much as we can onto our GitHub, but also our front-end library components as well. And all of these are available via an Apache 2 license, which means you can use them however you wish, just give a citation. And in addition to the PDBe-KB, we also have our core PDBe API. So if you're more interested in getting access to a certain subset of entry data, i.e. the structures which individual scientists have solved, then you can use the PDBe's API for that. And then, of course, the AlphaFold database as well. And I'm sure you've got to talk at some point going into the details of the AlphaFold database. Now, all of that said, if you just want the raw files, we also have our FTP server, so our transfer protocol server. And this is a great place to go if you just need an MMSIF file, a CCD file, You just need the raw data itself, basically. This is the place to go. It's great for programmatic access, and we also have some benchmark data sets on there as well, which have been curated for different purposes at the PDBe. So the overall architecture and archival process, I mentioned this already in passing, but we'll take in computed models. These can be from the 3D beacons, but also from the PDBe archive more generally, and we'll ingest them into our knowledge graph here over sort of in the center here, the hub of the spurs, so to speak. And this is where we can then retrieve data from the PDBe point of view, but also AFDB, the aggregated views of proteins. So this is fundamentally what the PDBe knowledge base is at the moment from a web UI point of view. But we also have our aggregated views of ligands, which have just come online recently, but also our aggregated views of complexes, which is something coming up later, and also aggregated views of domains as well. So yeah, keep an eye on that space. Okay, so if anyone would like to follow along, I've got the link over here to the PDBe-KB website, or you can scan the QR code if you'd like to follow along. Absolutely no need to. I'll be walking through the process, just having a look at the web pages. But you'll be taken to the search bar over here, and this is where you can look for, where you can search for your proteins by PDBe accession, so the 4-letter code, or uniprot accession as well. And once you make search, you'll land on the summary page over here, which will look something like this, where you have the name of the protein in the top left over here, as well as the G name, the organism in which it comes from. So in this case, I've searched for an ATP-dependent kinase, and you'll get some summary information in terms of the structures that are available within the PDBe archive. So in this case, for this hexakinase, we've got 4 structures within the PDBe archive. This might have increased since I last took the screenshots. You've also got a 3D viewer on the right-hand side there. It's referred to as a MolStar viewer. So if anyone's used PyMol before, this is PyMol Chimera. This is similar to that, but it's all web-based, so you can load it into a browser, and you can get really nice figures, publication-ready figures from that as well. So this gives you a real overview of the structural context for this specific protein across the PDBe archive. And in the other tabs, this is where we're really trying to achieve that data enrichment side. So of course, we'll show the 3D view of the protein over here. This will be the representative structure, which is usually a structure which has very high resolution, or the highest resolution given all of the PDBe structures within the archive, as well as maximizing for sequence length as well. And then along the bottom here, we're also providing a sequence level view as well. So this is where we can add additional annotations from our 34 different collaborators, or which of those 34 collaborators are providing sequence level information, which is worth displaying over here. I'll talk about that more in a bit. Additionally, as well, we've got a ligand tab as well. So across the PDBe archive, there'll often be lots of different small molecules bound to proteins, and going through every single entry one by one would be quite cumbersome. So what we try to do here is collate them all together, so you can see the different molecules, the different small molecules which are bound to this particular hexakinase across the entire PDBe archive as well. Again, I'll be talking about this more in a bit more detail later. We have an interactions tab as well. So this is really giving you a view on the assembly composition. So in this case, we've only got one other protein that we find in complex in the PDBe archive. So this would form a homodimer over here. But the 2D sequence view, the 1D sequence view, is really giving you an idea of the heat map. Where in the sequence is this protein here making the interface? And then finally, the annotations, and this is where all 34 collaborators' data comes together to show you an overall view of all the different things. We've got things like the domain annotations, the actual ligand binding sites, the actual interaction interfaces from the previous 2 tabs, but we might also have things like predicted ligand binding sites as well. So this can be quite useful where, well, not every single ligand which could bind has been solved within a structure. And I'll be talking about that in a bit later. So as I alluded to earlier on, a lot of this data comes from a graph database. If you want to write your own queries, then you can download the entire graph database. I believe it's just over 100 gigabytes at this point, so you might have to use an institution's HPC or cluster to make large queries. But yeah, this is probably the most comprehensive place to go, because having this node-based structure, this node and edge-based structure, can allow you to write queries not only on descriptors for the entity, but how entities connect with each other as well. And you can go all the way down to the residue level with the PDBe-KB's graph database here. So as I said, all annotations are over on the graph database. We've also got the FTP server as well for the raw files, and we also provide an updated MMSIF format. So the MMSIF format is the file format which pretty much superseded the PDB file format. And the updated MMSIF file format is where we're trying to integrate annotations from UniProts into the structure file. The idea there is that one of the big benefits of using the updated MMSIF file format is that we unify the residue numbering across every single entry. So where depositors, so the structural biology investigators who solve a structure, can give their residue labeling any labeling they would like, as well as the chain IDs as well. They can call them anything they like. We try to sort of standardize those, unify them across the entire PDB archive. I think that's it for this slide, yeah. Okay, so I'm going to go through a few use cases of for the PDBe-KB. This is by no means an exhaustive list, and I'm sure you'll find new creative ways for using these data and the web pages yourself for your own work. But hopefully by the end of the walkthrough, you'll be able to describe the mission of the PDBe-KB, and most importantly, identify some of the tools which might be useful for your own work. By the end, you'll find out how you can anyway identify some sequence and 3D level structure information, navigate the PDBe-KB's web pages to find those annotations, but also query the graph database via the aggregated API that we have to get those additional annotations, which you can add to your own pipelines, whether for data analysis or write more specific queries as well to get the exact data that you need. So here's the link again. I'm going to be going through the specific tabs and the data that's available there, but if anyone wants to follow along, feel free to click on those links. I'll keep them on screen for a second. As I said earlier, the search bar over here will only accept the UniProt accession or the PDB ID itself. But as I'll talk about, you can actually access it from the PDBe-search page as well. Okay, so once you make your search, you'll land on a page like this. It's the same one as we were looking at earlier. This time I'm searching for a serine threonine protein kinase, PLK4, and the first line will be, things like the gene information. So the gene name, PLK4. We also reference or link to the Enzyme Commission website as well. So in this case, it's an enzyme. So you can find out more details on how the enzyme works, the biochemical pathway it's involved with, and also some of the disease implications that this one has as well. We've got the organism name here too, so Homo sapiens, this is from a human. And often the protein they're referred to as different, got different names depending on the discipline. So we'll also write the synonyms here as well. So you've got that information if you're looking for a particular protein. It might be referred to as something different depending on the discipline. So we've got those different names over there. We also link to UniProts as well, and they also include the UniProts description for the protein's biological function. Now on the right-hand side over here, this is the 3D sequence viewer, and one of the reasons I'm picking this as an example is oftentimes you'll have your entire UniProts sequence, the entire sequence of the protein, but within the PDB archive, researchers might have only solved certain sections of the entire sequence. There could be a few different reasons for that. Maybe this represents only a single domain, so this is the only part which is of interest to research. Sometimes these can be long, intrinsically disordered regions as well, so they might be quite difficult to get crystals or cryo-EM structures for. So they might be, they might not be present within the PDB archive, but it's at least there. And we refer to these as different segments, basically. So in this case, we've got 3 different segments, one over at the N-terminal region, one sort of in the middle over here, and one at the C-terminal region as well. So in terms of those structure annotations, clicking on that tab, yeah, you'll get this the sequence viewer over here, and this is where we're providing on a residue level anyway, things like secondary structure variations, so where are the alpha helices, where are the beta sheets, where are the loops. From our collaborators who work with us on the PDBe-KB, we've also got our flexibility predictions as well. So this could be quite useful for looking at whether a protein has any intrinsically disordered nature. So is it moving around a lot? Is it very dynamic? Does it not hold a particular secondary structure for very long? Topology annotations as well, where it might exist within the membrane. Early folding residue predictions as well, and then our domain annotations too. And these come from Interpro, CAF, SCOP. Can be really, really useful for looking at proteins that maybe share a domain with other members, with other proteins, but maybe the function hasn't been well characterized. So you can use these domain annotations to get an idea of, if you've got a particular protein of interest and it shares some domain sequence similarity with another one, like maybe this one over here, you can try to predict maybe what it's doing as a result before going down and trying to do some wet lab investigations with it. And the very final channel over here is the other structures, and this can be where we'll have the AlphaFold structures, maybe from Swiss model as well. This can be really, really useful because they can fill in the gaps. In this case, we've got a region somewhere between residue 300, residue 600 over there as well. And this has not been modeled yet within the PDB archive, but you could, if you wanted, you could go and use those predicted models to try and predict what's the what the sort of intervening structure is. You might find that it is really, really tough to predict. It might be very, very intrinsically disordered. Now the annotations tab, yeah, this is where we've got all of the annotations collected in total. And this goes, yeah, everything from things like predicted functional sites. This will really depend on what the function is and what the collaborator is predicting. Predicted PTM sites, so PTM sites for post-translational modification. These can be things like meristylation, ubiquitinylation, phosphorylation. So it really depends again what the name is. But it can be quite useful to see, okay, maybe here this protein is being phosphorylated quite early on, somewhere towards the N-terminal region, and then there are a few other sites, maybe they're over here and over here and here, which could be important for, maybe modifying its function as a result. Predicted ligand binding sites, this is often a really useful one to look at in conjunction with the actual ligand binding sites as well. So where the predicted ligand binding sites might show you a kind of a greater distribution in its heat map, you can compare that against the actual ligand binding sites as well. So where the ligands are found within the structure. You might find there are some regions where there's a predicted ligand binding site, but no ligand has been solved yet with that. No structure has been solved yet with that ligand bound to it. So this would be quite a useful thing to inform for maybe drug discovery as well. And then of course, yeah, the other annotations from the previous tabs as well on the structures. We've got the domain annotations over here too. Yeah, so as I mentioned a little bit earlier, you can also access the PDBe-KB from the PDBe websites as well. And this can be quite useful because the search function is a little bit more comprehensive. So this is where you can search for the PDB code directly, the protein name, the ligand name, gene name, or the uniprot succession as well. If you want even more control, we also have this advanced search feature over here. So this is where you can start to include things like Boolean logic. So do you want, structures with a certain residue length, or a certain resolution cutoff, or do you want to exclude certain organisms, include others? You can get really granular with the advanced search feature over there. So it gives you a lot more control. Once you make that search, yeah, you'll find all of the results over here. So for my search that I made in this case, I got 344 results. And this 344 will represent individual proteins that have been solved and then deposited. So these kind of, you can think of these as like individual experiments, individual structures which have been solved and deposited. And you can navigate over to the PDBe-KB website using either of these two links over here. In this case, for this protein, there are two links, because as you can see in the image on the right-hand side over here, you've got one protein in green and then another one, it's probably not too clear over here, in cyan. So because there are two proteins that have been solved within the same complex here, you're going to have two different links, one for each protein within the complex. If you do want to see just the macromolecules, then you can click on the macromolecules tab, and because there were only two proteins that were yielded from this search, you'll only get two macromolecules roles. But nevertheless, you can access the PDBe-KB from this page here as well. So here are some questions that that I've come up with, which you could use the PDBe-KB to answer. Again, there are only 4 of them here, so there's by no means an exhaustive list. And I'm sure you'll come up with much more interesting ones than the ones I have. But number 1, you might have a question like, what ligands does my protein bind? Again, quite an important question for any drug discovery, drug design. And you would initially come here via searching the PDBe, as we talked about a second ago, or go to the PDBe-KB directly. And once you're on the summary page, navigate over to the ligands tab. And this is where we'll initially show a tile view along the top here, where we'll have all of the different ligands which have been solved along with structures that map to this target protein over here, this query protein. So we've got things like LPA, NDP, NAP. So in this case, they're all labeled as cofactor-like. And this can be super, super useful, because if you know something about the biological function of these ligands, well, number 1, this can help your research, what biological question you're trying to answer. But again, from a drug discovery, drug design point of view, if you know these are cofactor-like, then you know anything else binding to this site is having potentially some sort of cofactor-like effect. Now, on the residue level as well, scrolling down on that page, you're going to get this track view over here, and you can start to see where in the sequence those ligands are bound. And what I've done is I've labeled here, of course, if you click on that tab, that will create a drop down and you'll see where every single ligand is binding. There were quite a few in this case, so I sort of collapsed them all to get this summary view instead. But there are 2 sites in this case where the cofactors are actually binding. We've got one site at around residue 20 and another site around residue 50. And then the others will represent potentially maybe an active site somewhere else, or maybe some sort of allosteric site as well. Okay, so second question on my list anyway is what complexes does my protein form? And again, you can access this either by going from the KB page directly or from your PDBe search. But nevertheless, head over to the Interactions tab. And this is where you'll get listed all of the other proteins, all of the other macromolecules, which have been solved in complex with your given query protein. So in this case, my query protein was that serine threonine protein kinase. I can see 4 other proteins that have been solved LK4 protein. And in this case, they're all pretty small proteins. And if we move on to the next slide, scrolling down, we can see exactly where within the assembly those proteins have been, or exactly where within the sequence these proteins are making an interface with the assembly. If we look through, they're actually very, very, very consistent. There's only one down here which is not making those interactions at around residue position 50. So maybe it's making a slightly weaker interaction, or maybe these contacts over here are compensated for in some way. And by looking at the names of the entry as well, we can also see what kind of effect they might be having. So in this case, it's a CAMP dependent, so it's a cyclic AMP dependent protein kinase inhibitor, and it looks pretty consistent. These 3 over here, these are all having potentially an inhibitory effect, but the top one over here is not. So there might be something interesting to go and investigate over there. What's the difference between this one and the kinase inhibitors? Are they just different names, or is this maybe having something which is not an inhibitory effect? So thirdly, this is something that I've been working on since I joined a few years ago now, which is trying to identify the different conformational states that proteins adopt within the PDB archive anyway. So proteins are very flexible molecules. They move around a lot. They're not static. But within the PDB archive, we basically have either ensemble models from investigations like NMR, or maybe some some time-resolved crystallography. Too many of those, but theoretically you can get that data, or cryo-EM as well, which can also theoretically give ensemble models as well, within the constraints of the electron density anyway. But nevertheless, we get those static models, and because of the high redundancy of some proteins, proteins with a given sequence that is the same, but many individual structures which have been solved independently, you can start to find where the differences are within the conformational states. And on the summary pages, there's a button on the bottom left which says 3D view of superposed structures, and by clicking on that, you'll see over here the different clusters that we predict. Now, the clusters themselves will not be de facto conformational states. To really define something as a biologically important conformational state will require more than just looking at the atomic coordinates. But nevertheless, we are clustering them by those atomic coordinates. So here the clusters, cluster 1 and cluster 2, the structures within them will be more conformationally similar to each other than they will between themselves, or the structures will between the clusters. And in this case here, we're looking here at segment 3. So if you were to click on that button, once the viewer opens up, you'll be able to see all of the segments there. And these refer to the discontiguous segments within the UniProt sequence in which protein structures have been solved within the archive. And there's this pretty large hinge region where the beta-pleated sheet is either in this kind of upwards conformation in the green structure, which would represent cluster 1, or the kind of downwards folded structure, which is the case for cluster 2 over there. Now, it's been out a few years now, but we can also stream in the AlphaFold structure as well. And because the AlphaFold structure represents the entire UniProt sequence, or the entire protein sequence, you won't only get segment 3, but you'll get the rest of the structure for all of the other segments as well. So it'll be quite useful for seeing how they might relate to each other if and when the structure gets solved. And when streaming in the AlphaFold model, it can be quite useful to look at the RMSD. This stands for root mean squared deviation. It's kind of like an atom-atom distance comparison for all of the available atoms that you can compare. And in this case, the RMSD between the AlphaFold model and the chain in cluster 2 over here is only 1.21. So it's a very low RMSD over there, relative to 12.38. So in this case, the AlphaFold model, interestingly, has adopted the conformation of this cluster, of this conformational state over here. But what you can see in the archive is actually there is this other conformational state, which is accessible, which is stable. But the AlphaFold model, at least the top hit, is not presenting that because it is also just a simple snapshot. It can be important to have a look at the PAE as well, so the position alignment error, as well as the PLDDT2. So just check, is the AlphaFold model, can you confidently say, where it's predicting the atomic coordinates? And then finally, some more compound questions, pun unintended. So some more mixed questions where you might have things like, does ligand binding affect protein conformation, something like that. But we also have the 3D view of superposed ligands down there as well. And by clicking on that, this is where we strip away all of the protein structures, but the top hits, the representative structures for a particular cluster. Now we're looking here at a slightly earlier segment of the N-terminal domain. And we're only showing here the representatives for cluster 1 and the representative for cluster 2, and we can see where in the protein the ligands are binding. On the right hand side here, you can toggle on and off the different ligands which have been solved for all of the structures for this protein in the PDBe archive. And we can see here, yeah, there's this kind of internal binding site. I imagine here this is more than likely the site of phosphorylation, considering it's the kinase. There might also be some sort of allosteric site over here on the right-hand side too. Now, you can also access this information programmatically as well. Regarding the 3 questions we've just gone through,
आवाज नहीं आ रहा है ना? आवाज आ रहा है? आवाज आ रहा है? मुझे नहीं आ रहा.
use, some Python scripts or R scripts to fetch the data as well. The only things you have to specify are potentially your PDB ID, the entity ID as well. So if there are multiple molecules within your MMSIF file, you want to specify which one you want there as well. So PDB ID might have several ligands and other macromolecules in there. If you want the bound ligand interactions as well, so where in the protein, which residues in the protein the ligand is actually making contact with, you would start off with this stem for the URL, and then also paste the PDB ID, the chain ID, and then the specific residue ID over there that you would want to. Obviously, I won't go through all of these, but this is the idea basically, is that all of this information is accessible through our Graph API or aggregated API, as we're referring to it these days. And if you want all of the annotations for a given PDB accession, we also have the fun PDB API endpoint as well, where you can just plug in the PDB ID, so the 4-letter code, and that will give you all of the annotations. These will be returned in JSON formats, so they work quite nicely across different tech stacks. So yeah, if you wanted, I'll just list these links over here in case anyone wants a reference to them. We could recap. So this is the sites of the PDB knowledge base. If you want more information on what the different API endpoints are and what data they provide, this is the endpoint documentation over there. The FTP area is where you can find all the raw files. This is something I've been working on. So the clustering results is something you can access. Just as an example on how you can navigate the FTP area, because it's fundamentally just a file server. And then we've also got some benchmark data sets as well, so we can also search for benchmark data sets there as well. You can download benchmark data sets there too. Okay, so I think a bit under time, but yeah, just to recap really, the PDBe-KB aims to provide this fair infrastructure, so findable, accessible, interoperable, and reusable. As I've been talking about, you can access these annotations via a browser, but also programmatically using those API endpoints. Hopefully, this will provide you guys, the researchers, with the relevant structural information on a residue and 3D level. It should give you quick navigation through the websites, both at a glance, and then if you need to get that data for your own research, then you've got the API endpoints and the entire graph database as well to make your own Cypher queries. So I'd like to thank the funding partners, EMBL itself, UKRI, Wellcome Trust, European Union, and Google DeepMind, and then our collaborators as well for sort of the WWPDB, of course, there's the 3 sites working together, 3D Beacons network as well, SciTech, who help us develop, help us develop the Molstar viewer, which is really fantastic. The Elixir groups as well, working groups, 3D Bioinfo community, and then again Google DeepMind as well for working with us on the AlphaFold database as well. And I'd like to thank the team too. So we're just over 30 people now in total. It's a fantastic team. Everyone's working really hard to make sure the data that we distribute to the community is of as high value as possible, really. And yeah, I think I'll move on to questions. So a bit under time, but
yeah, thank you, Mr. Joseph.
Now would be a good time. Yeah.
Yeah, thank you very much for the excellent presentation on PDBe-KB. So this is very clear and informative overview of all the resources of PDBe-KB and all the functionalities and the integration with the other databases. Everything is very clear, and I hope this session is inspired all the attendees to explore these resources further and utilize these resources in their work. If you have any questions in between using these resources, you can directly reach to the EMBL-EBI, PDBe-KB staff, or RSG-India. We are gateway to the EMBL-EBI, PDBe-KB. And yeah, it's time for the questions. Audience, if you have any questions, you can unmute your mic and you can ask your questions. Yeah, Garima Metal, you can. Yes, Garima Metal, you can unmute your mic and you can ask.
Good afternoon, sir. First of all, it was a great session. Actually, currently I'm pursuing my bachelor's in biotechnology. I wanted to ask if, apart from that, I want to like become a bioinformatician. So, what skills should I like acquire besides my academic skills to achieve my goal?
Oh, that's a good question. Yeah. So if you're doing biotechnology at the moment, as far as I'm aware, yeah, biotechnology is often one of those degrees which is quite diverse in terms of like depending on the institution you're doing your degree at, exactly what you'll be studying will be dependent. So I don't know exactly what's in your course, but I would say go and learn some programming if you're not doing that already. Probably the most useful language for most bioinformatics is going to be Python these days. But learning some R can be useful as well. I'd also recommend getting some machine learning experience too. I think that's something which is going to just get more and more important. Getting familiar with different data formats as well. It's really going to be dependent on what type of bioinformatics you're interested in. Is it structural bioinformatics, genomics, multiomics? It is bioinformatics is somewhat of an umbrella term where there's so many different avenues you can go down. But yeah, I would say depending on where you are in your journey, get some Python skills. Yeah, find a project or some sort of research tool that you've either been working on. You can get a GitHub repository going, and make that available. That'd be my advice.
Yeah, thank you, Mr. Joseph. And Dr. Manisha will have one question. Ma'am, please. You can.
Hi, Joseph. Thank you for a great talk. I actually just wanted to ask a simple question. So you were showing us a place where we are able to find different conformations of the same protein. Would there be any information available about the energies of these conformations?
Oh, it's a very good question. Yeah. Not from the PDBe side. I mean, one thing you can take almost as a given is that within the within the archive, what you're getting are the low energy conformational states. So the conformations which are being predicted here will be those which are pretty stable because they were able to either be crystallized
Yes.
or frozen on an EM grid.
Yes, that we understand. I was just wondering whether changes in conformation and binding of ligands, would they show us differences in energy or even binding energies for ligands if that information is already available? That's all.
Well, so if it's for conformational binding of ligands, I know we at the PDB have a PISA. I'll have to double check whether we provide the ligand information as well, because I think PISA is less reliable on ligand binding metrics.
Okay.
It's much more reliable on protein interfaces. Yeah, no, it's a fantastic question. Yeah, okay. Yeah, but fundamentally there will be differences there. Yeah.
Yeah, thank you, Dr. Joseph. We have another question like, how can PDBe-KB help researchers analyze structural variations and mutations using PDB codes?
Using PDB codes, yeah. Well, the whole one of the big ideas with the knowledge base is because you are integrating lots of different independently solved structures across the archive. Sometimes there will be either intentionally, so some of there will be mutations which have been introduced to improve the stability of the protein, or to make sure that it could get a crystal structure for it. Maybe to make sure that they want to get a substrate bound as well. That can often be, so if there's like an active site residue, which is important for catalysis, you might have to mutate that to maybe an alanine, let's say, just to sort of inactivate the enzymatic activity of it, just so you can then get the structure. I mean, that said, if you want to do a more comprehensive mutation analysis, you'd have to go with the very well characterized proteins. The mutations themselves will be captured. So, for example, if there's a given uniprotic session, so you're looking for a certain protein, that will also include any mutation, any proteins with mutations in it as well. It's eventually, if you keep adding more and more mutations, then it becomes a different protein. So if there are just single point mutations, they will be captured in here. And they basically appear as like a little red line on those sort of sequence viewers there. Yeah, I hope that answers your question. They can be a difficult thing to probe systematically.
Yeah, I think, yeah, you answered that question, Dr. Joseph. And another question we received through online registration form. So is there any way to validate a model structure through PDBe-KB? Is there any resources available in PDBe-KB that we can validate?
To validate the model, absolutely, yeah. I mean, if I go to maybe the PDBe website more broadly, I don't want to jinx it by doing a live demo. But if I was to search for, maybe call it 180p, open up in a new tab over here. I believe the metrics you're probably looking for will be on a per entry basis. So this is where we've displayed it over here. This is referred to as our entry pages. So this is giving you a load of info. So this is not the KB itself per se, but this is where you can get things like experimental and model validation as well. There'll be a lot of things like clash score. This is where you might have atoms clipping into each other. You want to make sure that's as sort of as low as possible, as well as the Ramachandran outliers as well. So this is where the bond angles within the backbone might be non-optimal. Of course, that might match the experimental data very well. So maybe there are situations where Ramachandran outliers are realistic, but sometimes they might just be maybe sort of modeled within the protein density, but maybe they should be in a slightly different position. So this is probably the place to go for a per entry experimental validation. But it's a good point. Yeah, I mean, collating all of that data across the archive is challenging because we have fundamentally pretty much three different experimental techniques which come together to describe the PDB archive. Primarily nuclear magnetic resonance, so NMR, X-ray crystallography, and cryo-EM as well. So finding unified metrics which take these quite independently, quite conceptually independent views of structure validation and putting them together can be a real challenge. But nevertheless, looking at the model coordinate is pretty valid, nevertheless.
Yeah, whatever we look, we have to look for PDBe-KB now. Everything is integrated here. Yeah. So
and you can go back and forth as well. So you can go from the knowledge base to the specific entry pages as well. Yeah.
So we have another question. So it's like, if protein needs a folded structure to function, why do many exist in a state of disorder? How does PDBe address the challenge of showing protein dynamics beyond static structures?
Yeah, really good question. Yeah, so so the reason that might be is sometimes they'll either need an interacting partner to adopt a stable conformation, but fundamentally it's going to be the sequence of the amino acids which are resulting in the protein not being able to form a stable alpha helix or beta pleated sheet. There are several examples where you will need a separate binding partner, and in cases in which those structures have been solved, they will be on the protein data bank. That said, you then won't be able to see the intrinsically disordered view. The only situation you might is with any NMR structures, because those can much more reliably capture intrinsic disorder. But something like crystallography and cryo-EM, they do struggle to capture very, very dynamic and mobile regions of proteins. The other place to go as well, and you can integrate this in with like the conformation viewer over here, is when you have a look at the AlphaFold model. Sometimes low PL-DDT can be a good proxy for intrinsic disorder as well. And because the AlphaFold model is giving you effectively the entire protein structure, you can start to see where maybe domains are connected by a disordered linker. But those disordered linkers can be very, very important for the protein's function. You think of like the NP, the FP nups within a nuclear pore complex. It's almost like their intrinsic disorder is like fundamental for the function of the nuclear pore complex. But yeah, they're very difficult to capture from crystallography anyway, because you need a stable structure, you need a crystal.
Yeah. Actually, and we have so many like the fresh graduates now, like they completed their graduation and masters. Now they are asking the same questions like, so can you please share your career journey? And some of them are completed masters in bioinformatics, masters in biotechnology, biochemistry, and they want to just want to jump into bioinformatics. So if you have any tips to them, like uh, somebody asked like start career in bioinformatics focused on proteins, and what are the essential skills needed to contribute? So could you share some?
Yeah, I mean, it's going to depend on the domain you're looking for, because as I said, there was a question earlier about getting about getting bioinformatics skills. It's a field which is it's a very fast moving field as well. So I'd say keeping your finger on the pulse with what new developments are coming around, and trying to find, from a careers perspective anyway, maybe trying to find your niche, trying to find your specific interest. Because there are lots of tools coming online all the time for trying to answer different research questions, trying to solve different things within bioinformatics. So yeah, identify what is really of interest to you. The same advice that the earlier question had as well. Get a GitHub repository going. Put your code on there. That would be my advice, really. It's find a niche and get your work online. And if it's something really useful and important too, people will use it. I mean, it can be a bit of a marketing tactic when putting things on GitHub. So putting on GitHub nevertheless though, at least you can find you and find your tools useful.
Okay. Yeah. And Dr. Joseph, do we have time to take some few more questions?
Of course, yeah. I've got until 12:30. So.
Yeah. Dr. Ashwini Mishra, you have a question you can ask.
Hello, am I audible to you?
Yeah, you are. Yeah.
First of all, thank you very much for such an informative session. I've been working in the field probably for the last 7 years, and I've been doing like lots of molecular docking and other work. The thing which really is the reason for pain for me is I'm into pharmaceutics and I'm into biopharmaceutics. So, in pharmacy also you have to go for, molecular optimization, new drug target searching and all. But when it comes to molecular dynamics simulation, it's really tricky because the only free thing I know is GROMAC, through which you can go for doing molecular dynamic simulation. Apart from it, and for that particular thing, I need to ask for the help from many people over here. Is there any other way through which, like, molecular dynamic simulation can be done without script?
Yeah, so are you asking on the software side using GROMACS?
Exactly, yeah.
Yeah, I mean, I know there's Amber as well. That can be another option. There is a database called MMDB. So MDDB. Sorry. So that's the Molecular Dynamics Data Bank. That might be something worth looking into. I know it's a database which is coming online more and more. We're collaborating with them on that. And the idea there is that will be similar to a lot of the things EBI does. It's going to be an open source database for molecular dynamics simulations. That might be quite useful for your work, because the molecular dynamics simulations can be incredibly expensive to run, and you often only get maybe a few picoseconds captured. One thing you could do is try and integrate experimental structures with your MD simulations. Almost waypoint them, so map where known conformations have been solved in the PDB archive onto your simulations. But no, the MD simulations are incredibly expensive. I've never had to run them myself, but yeah, there's some people in the group. agree too.
molecular permeability is being simulated
I
by
think
using machine learning tools. And people create artificial membrane, and the permeability of the nanostructured crystals or probably
yeah.
particles are being done. Could
Th-
you please give some light on this?
Dr.
How it
Ashwini
can be done,
Mishra,
please?
thank you for the question. I think we can continue this question later on. Just we can focus on PDBe-KB right now. And next we have another question from Dr. Sudhanshana Ganguly. You can ask now. Yeah.
Sorry, this isn't a question. Just to add on
Oh,
to, so I also
hi.
work at EBI, and just to
Yeah.
add on to Joseph's comment, it's and regards MD simulations, it's extremely important to be aware of which starting structure you're using when you start any MD simulation, and maybe we can look to conduct a talk or seminar in future about how if you have 20 PDB structures for the same protein, which one do you start your simulation with? There are lots of um, considerations that you need to have. And also, as Joseph mentioned, MDDB is going to be a great resource where you do not need to perhaps run the entire MD. You can, you know, go, and if there's already an MD simulation that's already out there, you can probably download the data, look at certain frames that you're interested in. So it's really going to reduce the computational power you need. As far as I am aware, Chromax is the only free software that's available right now in the market. Helps you with your MD simulations. Amber is paid, so not everybody may have access to it. But yeah, that is something maybe we can, RSG India and EBI can probably look into in the future.
Thank you very much.
And yeah, thank you, Dr. Sudhanshana. So due to some technical glitch today, we are accommodated up to 100 persons only. So please keep registered for the upcoming sessions to accommodate your spots. And thank you very much, Joseph, for your session today, and it was very helpful. And
No, thank you so much. It's a
yeah,
pleasure.
I would like to welcome Nikita to give your concluding remarks. Yeah, Nikita. Yeah.
Thank you, Govind, for providing me this opportunity to just lead the session. So the session has been very, very insightful because these are the resources. I think, like me, most of the audience have never explored all these things. So thank you, Joseph, for just bringing out this picture to us in front of us, because being a researcher, it is very useful. Like these resources are with us, but we are not aware of it. So that insightful knowledge is very important. I think the researchers who are just stepping into this world of exploring computational biology or computational chemistry, they'll find it very useful for them. So thank you, Joseph, for providing this wonderful, wonderful journey experience of PDBe-KB. And thank you, Sudakshana, for adding to it, because sometimes we miss some points that are all asked in audience, and we sometimes don't know how to answer them. But Sudakshana, you did a wonderful job to it. So I really would like to thank EMBL-EBI and PDBe-KB for just bringing us these wonderful illustrators to us for this session. And I would really like to thank ISCB-SC for providing this platform for our platform in association with RSG India, so that we have opportunity to bring these type of topics for our audience, because these topics are sometimes missed and we are running in without information. So thank you, RSG India and ISCB. And for any session to be very helpful and engaging with the audience is very important. So I think I have seen from our that the audience is intact there. So thank you, audience, for joining the event and making it more interactive, because the questions were really very engaging. So thank you, audience, for joining and staying updated. And thank you, RSG India, RSG India core team, who have managed all these things very well, because it for bringing a single event takes a lot of effort of a core team. So entire team of RSG India, along with the volunteers who have put a lot of effort in bringing this platform online. So thank you so much. So this was the first wrap up of the session of this Edu series. We'll be having more sessions like this in future. So stay connected to RSG India and its website. We are available on social medias like LinkedIn, Twitter, Instagram. Please stay tuned to us. So all the you can scan the QRs on the screen as well. The feedback forms will be updated in the chat soon and will be shared to you in the emails as well. So don't forget to give us your feedback because these feedbacks makes our other events like more benefited for you. So last and not the least, I would like to thank our faculty advisor as well for keeping us like motivated to have these sessions again and again. So thank you everyone for
Nikita, sorry to interrupt you, but we forget to take a group photo. So I would request
yeah.
everyone
At
to,
the end, I was about to ask for it.
yeah.
So before leaving the session, please everyone switch on your cameras so that we can have a very like we have a last photo for the session, and it is the first session Edu series, so it should be there. Who else is taking the screenshot? Please do it.
It's done. Thank you, everyone. Yeah, thank you everyone for joining. The screenshot is taken. Yeah.
Thank you everyone for joining, and thank you Joseph again for such insightful journey. Thank you so much.
Thank you so much to the organisers for having me. It's a pleasure. Thank you so much.